So good afternoon. We're going to try this again, and hopefully this will be the last time that we have to resort to a recording that happens later in the afternoon and is not the actual recording that happened in lecture. And hopefully the live stream will actually work as well. So we are the same people. I'm still Professor K. We still have our 7 teaching assistants and interns. Their names are shown for you here. And oops, their pictures are shown for you here. And actually, I've already made some marks on this screen, so we're just going to go with it. Haley and Chenan, and I think, no, so Haley and Chenan here will both be doing virtual sessions for you all. And they will give each 4 hours of time per week. So you'll have 8 hours of virtual sessions. And then B. Chen, another graduate TA, will provide an additional 4 hours, but these will be in person. And each of the undergrads will each provide 2 hours of in-person. So another 8 hours here of in-person. So 12 hours of in-person time, 8 hours of virtual time. The virtual slots, we will post links to those sessions on Brightspace for you, so you'll have direct access to those. And the in-person hours will actually happen in the BRC. And so here's that calendar of times that's posted for you on Brightspace, including the virtual or in-person times. We're the only class that'll be using the BRC, and that is in Lily B401. That'll start next Monday. And so all the assistants that are down there will have keys, so they'll be able to let you in. And I ask that everybody cooperate. Please disinfect the region that you're sitting at when you leave. Make sure your mask is on at all time. And hopefully we don't run into this, but if we get to the point where we get too many individuals down there, we may have to resort to a sign up. I really hope that that doesn't happen. But I'll be in touch with the TAs weekly and we'll see how that goes. Okay, so let's talk about cell biology. That's what we're all here for. And the first thing I want to familiarize everybody with is this idea of sizes. Now, this is a schematic. It's available to you in your lecture notes. You already have access to those. You should also look at these 2 figures in your text that I have highlighted here for you. And this is where we're going to spend the bulk of our time in cell biology. So a couple things, and I'm going to switch back and forth between this PowerPoint lecture as well as some handwritten notes for you during the course of this lecture. So with regard to sizes, I want to think about a few things here. And the first one that I want to think about are where we're going to be spending our time. We're going to spend our time in the millimeter range, which is 10 to the minus third meters. We're going to spend our time looking at things that are micrometers in size, and that's 10 to the minus 6 meters. And we'll look at things that are a little bit smaller in the nanometer range at 10 to the minus 9th meters. And you're all familiar with these 3. These are all SI units. We will go down 1 order of magnitude smaller to 10 to the minus 10 meters. And this is something, it's a non-SI unit. Structural biologists use this, and this is the angstrom. And so this is where we're going to spend our time. And if we go back to that PowerPoint figure, you'll see that here. So if we start kind of in the millimeter range, you're starting to see things such as large cells like a paramecium or a oocyte. In the high micromolar range, we start looking at cells that encompass the majority of our body, epithelial cells. And as we kind of dive a little bit lower, we start getting into some organelles like the mitochondria. We start to see that we're in the size range of bacteria and get into sizes of viruses once we hit kind of the mid nanomolar size range. Some larger components in the cell like ribosomes, we'll spend some time there, and actin, and so on and so forth, all the way down to the angstrom where we can see some atomic structure there as well as structures of some of these atoms. So what I want you to do is to study the figures in your books and in the lecture notes and to begin to get your brains around this and spend really some time here. So we're talking about 6 orders of magnitude that we're going to be covering over the course of this 13 weeks. And so that's what we're going to be dealing with. So there's a really neat slider that I want you to look at. Unfortunately, on the iPad, I checked it out in lecture and it doesn't work, and that's what I'm using to record right now is an iPad. But if you check out this link down here, scaleoftheuniverse.com, really cool link. It allows you to look at things over the course of 62 orders of magnitude. So it allows you to start at this very small, really the smallest thing known, which is quantum foam. And I asked in lecture how many people knew what this was, and I think maybe one, maybe two people raised their hand. I have no idea what this is, but what I know is that it's 10 to the negative 35 meters, so it's extraordinarily small. And that slider goes all the way up to the level of the universe, which is 10 to the 27 meters. So again, we're dealing with 62 orders of magnitude, right? So that's what we're looking at here over this course of from quantum foam to the universe. And if I were to ask you where you think cells fell in this continuum, I'll give you a second to make a guess on your own time. And I'm going to tell you that it's right here in the middle here. This is where cells are. Almost dead smack in the middle of quantum foam and the universe. And if you check out that slider and you put that slider right in the center of that website, you'll see that it'll focus right in on an oocyte, I believe. And you can click on the things in that slider. There's thousands of things that you can click on, from DNA to ribosomes to atoms, all the way up to large animals, and they'll give you some information. It's kind of fun to look at. Okay, so this is where we're going to spend the next 13 weeks. So what I want you to do is I want you to start to familiarize yourself with some organelles. So here's a cut through of a mammalian cell. And what we have here is we have the plasma membrane kind of in this somewhat darker sort of hot pink shade here. And the artist has cut the cell in 2 dimensions. So we have a cut going this way, we have a cut going this way, so that you can kind of see the inside of the cell. We can see the nucleus, and you see a lot of other structures, things that you're familiar with, right? You see things like this aqua structure here, which is a lysosome. And so start to familiarize yourself again with some of these organelles. And it probably is useful also to look at a plant cell as well. Plant cells have a few different things, one being the vacuole, the other being chloroplast that we don't see in mammalian cells. So familiarize yourself with that. Okay, so moving forward into our content for lecture 2, I want to start with the learning objectives. And again, just to remind you that these are available to you immediately. They're up for every single lecture right now in the lecture outlines, and then they become updated, or I shouldn't say updated, but available to you again as part of the lecture notes. They're on the very last page of the lecture notes, and you'll always find some sort of activity associated with the learning objectives. Here, the goals, once you go through the lecture today and review on your own, would be to explain something, and these are going to be the 7 boundary properties, and we'll discuss those. Use a formula to do some things, and that's to calculate surface to volume ratio. Explain certain things. Here's another explain and a draw. So all active things. And so these are great study guides to learn to figure out what it is that you should be able to do with the material that you're learning in this class. So this is the lecture outline for today. This is what we're going to be working on. We're going to start with the topology of cells, thinking about really the organization properties for these. So I'm going to go back to my OneNote and start jotting down some things that I want you to know. So firstly, we're going to spend some time looking at the general properties of ordered systems. Okay, and the first thing, the general property of an ordered system, of course, we start with order. And in the context of the cell, that order is really going to be maintained by a boundary. And there's two types of boundaries that we need to consider in the context of a cell. And in the case of cell biology, one is really intuitive. Probably one that you're thinking of is the plasma membrane. Right, and so what does the plasma membrane do as far as keeping things in order? If you think about it, the context of the cell, we have inside of the plasma membrane in the cytosol, we have typical pH 7, we have standard redox conditions, we have protein concentrations in the 100 milligram per milliliter range, all very different than what you'd find on the outside of that boundary, the extracellular space, which may have a very aqueous environment, may or may not have a pH close to 7. Redox conditions, very different. So we have the plasma membrane, and that helps to keep order within inside the cell. But one of the other boundaries that we often don't think about are the organelle membranes. So we go back to that lysosome inside of the cell. The environment inside of that lysosome is extraordinarily different than the environment just 5 nanometers away on the other side of that membrane, right? Inside of that lysosome is, don't put your hand in there, acidic, right? We're talking pH 4. And the only difference between the inside of that lysosome and the cytosol is a 5 nanometer boundary, right? And imparts order to that lysosome to have that low pH that's essential for degrading a lot of different components within the cell. Now, in order for all this to occur, and this shouldn't be a surprise to you, the cell needs to expend a ton of energy. And you'll see throughout the course of the semester that the majority of the time when I'm talking about energy, I will use a capital E. And it's using energy to overcome entropy or disorder. Right, entropy is S. Right, this is an easy one to think about, right? It takes a lot of energy to keep your dorm room clean, to keep the kitchen clean. This is the same, right? So a lot of energy to overcome the entropy in the cell. And so there's a lot of energy involved in maintaining that boundary. Okay, so what's the boundary like? So I want to think about this for a second. The boundary itself. So there's two things to think about with regard to the boundary. One is that its composition is primarily lipids, but we refer to this as a lipid bilayer. And as we're moving forward in cell biology, you're going to see things drawn at various magnifications, whether it's in your textbook or whether it's something I do. So as we're introducing the lipid bilayer, I just want to kind of highlight how you might see some of these things drawn. So in this case, the bilayer sometimes can be just drawn as a straight line, and I'm going to put dashes here to indicate that that goes in both directions, right? Because it's a continuous border. And in this case, actually in all cases, the lipid bilayer is 5 nanometers thick, right? But and let's go back just for a second to that PowerPoint drawing of the cell, right? That's exactly what we see here in this picture. So if you look at the boundary, the lipid bilayer, it's drawn as a single line. So that's exactly what the artist did here, was draw in the same fashion that I'm showing you here. But you'll see some images that are zoomed in. So let's zoom in a level. And what you might see, instead of just a single line, if you were to zoom in, are 2 lines to highlight those 2 leaflets. So the lipid bilayer is composed of 2 leaflets. And so if we zoom in, we can actually have a drawing where we see both of those. Of course, those are going to go on since this is a continuous sheet. And again, although zoomed in a little bit here, we're at 5 nanometers, right? And so you'll see many drawings in textbooks that use the 2 lines to indicate that. And then finally, we can zoom in even a little bit more to visualize some of the structures. And in this case, with the lipid bilayer, once we start looking at the structures, we'll see that what I'm drawing here in red are hydrophilic head groups that are going to be in contact with an aqueous environment, and those will go off in both directions. And then inside here, what I'm drawing in blue are the hydrophobic tails. And we'll spend some time looking at these in a bit more detail in a few weeks. But the bottom line here is we're still at 5 nanometers. We've just zoomed in a bit so that we can see a little bit more structure. Okay, so I just gave you a little bit of a kind of a tangent there on different ways schematically that we'll be looking at things in cell biology. All right, so the key here is that there's 2 leaflets, right? So you're going to have 2 separate leaflets that come together. So that's kind of the first important concept with regard to the boundary. The second important concept is that each of those leaflets is unique. So although in this image, let's see if I can get a highlighter for you here. I don't have a pointer. In this image here, I'm drawing both sides basically uniform. That's not at all how the cell membrane is composed. In fact, the outer surface is very different in lipid composition from the inner surface. So in this image, the region out here that I'm kind of generating X's for is the extracellular space, and then inside here would be the inside of the cell. And so I drew the bilayer as I did here. And so we have 2 lines. And each of those, the lipids that compose each of those layers are very different. And we'll learn about those. So we have very different lipids on each side. Or I'll call them on each face of that lipid bilayer. Additionally, it's not only the lipids that are going to be uniformly distributed differently between the 2 sides, but proteins as well. So in green here, what I'm drawing is just kind of a stick diagram of a protein. And what I want you to recognize here is that the extracellular side and the cytosolic side are very different. And so the 2 sides are not only different with regard to lipids, but they're also different with regard to proteins. And you will see my abbreviation for protein throughout this course being a P with a cross across there. So it's kind of a shorthand for protein. So very different proteins here. And also, put that back, also different in carbohydrates. So you won't find carbohydrates on the inner face of the membrane, only on the outer face. Okay, so the boundary has several properties, and I want to touch on some of these properties. But before I do that, let me just, only because it's fair that we looked at this in lecture today, I want to show you an artist drawing of a plasma membrane, 2 different ways to draw this that the artist is showing here. So in this case, we're kind of zoomed in pretty far, so we can actually see in the cartoon on the left, you see the hydrophilic head groups, hydrophobic tails. We have an aqueous environment on either side. And then we see more of a structural drawing on the right-hand side where the water is shown in blue. And we can kind of see the different atoms there. And then again, we have the hydrophilic head groups shown in red, hydrophobic tails shown in orange. So just a couple ways to look at that. Okay. All right. So 7 properties of cell boundaries. So let's spend about 10 or so minutes thinking about this. So boundary properties. Okay, so the first property is that it's continuous. Kind of intuitive, but this is despite a lot of complexity. It's not physically or anatomically trivial to be continuous, but nonetheless it is. And but you understand that because if the cell had holes, it's a cell without a future, right? So it needs to be continuous. So that one's fairly easy to, I think, understand and comprehend. The second one, something that you're probably also familiar with, is it's selectively permeable. So if this were a cell that has a boundary, had a glass boundary, it would also be a cell without a future. The cell wouldn't exist. And why is that? The cell needs to move stuff incessantly across that plasma membrane, right? This is one of the themes that we talked about on Monday about incessant change in the cell. So it's moving things like rapidly across the plasma membrane. Certain things cannot pass, like large globular proteins or certain ions, but other things like small hydrophobic molecules or dissolved gases like carbon dioxide and oxygen can easily pass. And so we talk about this selective permeability, and we say that the cell membrane has this ability to do this managed exchange. So it can bring stuff in, it can take stuff out, but not everything, only certain things, right? Again, really important, and we'll highlight that in other aspects of the course as we move forward. So the third boundary property is something that we already looked at, and that's that it's asymmetric. And in short, the inside face, as we just saw above, does not equal the outside. We have different lipid composition, different protein composition, as well as different portions of the protein on the cytosolic versus the extracellular side, and of course the carbohydrates as well. So we have this asymmetry going on. The fourth one is that the plasma membrane is a location of disequilibrium. Right, so we saw that when I mentioned the fact that you have this 100 milligrams of protein inside of the cell, and on the other side of the plasma membrane, you can be in an aqueous environment. So of course, locus for disequilibrium, right? You have a very different condition inside the cell as you do outside the cell. But there's two types of disequilibrium that we're going to talk about. The first one is chemical. And for this, I'll use an example of the calcium ion, but keep in mind that this is not the only chemical disequilibrium that's found in the cell, but it's an important one. In fact, in water, you have calcium at the millimolar concentration, but inside of the cells, you have calcium concentrations at the submicromolar concentration. What that means is that your calcium concentration is a 10,000-fold difference between the inside world and the outside world, and the boundary allows that to occur. Almost everything in the cell is out of equilibrium. If you find that anything in the cell is in equilibrium, it's likely a cell without a future. It's likely a dead cell. Okay, so disequilibrium, we have chemical disequilibrium, and we also have, and we're very interested in, electrical disequilibrium. So cells are going to maintain a voltage across the membrane, and you might be familiar with this with regard to neurons, but every cell has a voltage across the membrane. The membrane is like a capacitor, and the voltage across that can be tens of millivolts. So pretty small when you think about the millivolts, and that's going to be the difference between the inside and the outside. And it may not seem like a lot, but recall that the plasma membrane is only 5 nanometers thick. So I want to take you back to something a little different that you may have done in a laboratory at Purdue or maybe elsewhere. But for those of you that are familiar with gel electrophoresis, right? So you have a gel and you're separating something out on it. You're usually performing gel electrophoresis at about 100 volts, and your gel is usually about 10 centimeters, right? And so if we convert this to volts per centimeter, we're at 10 volts per centimeter. And I'll make the note here that this is gel electrophoresis. So that's the type of voltage we're dealing with when we do gel electrophoresis. And for those of you that have done that, you recognize that there's a lid on the gel electrophoresis tank. And that's because this 10 volts per centimeter is enough to give you a little shock. So you certainly don't want to put your hands in there. Now let's think about the plasma membrane and the voltage across that. So we're talking about tens of millivolts between the inside and the outside world, but we only have 5 nanometer thickness. We don't have a 10 centimeter gel, right? So what that relates to when we're talking about the voltage across the plasma membrane, we're talking about 100 kilovolts per centimeter, opposed to 10 volts per centimeter that you would have in gel electrophoresis, where you already don't want to touch that. So it is quite substantial across that membrane, and cells are going to use energy to maintain this voltage difference, and they're going to call upon that voltage difference to do work. So again, the plasma membrane acts as a capacitor or battery where it stores a lot of energy, and we're going to come back to that later on in the course. Okay, so that's disequilibrium. The fifth boundary property that I want to talk about is that the boundary is a locus for biochemical activity. So locus for biochemical activity. What do I mean by that? So 100 years ago, when people were studying cell biology, they thought cells were just bags of stuff and that everything in the cell was just diffusing in three-dimensional space. And we know that that's not the case at all. If that were the case, it'd be a cell without a future, because it would be extraordinarily difficult for two molecules in the cell to find each other if they were diffusing in 3 dimensions. What the plasma membrane does is it imposes some order. So there are some proteins that will be embedded in the plasma membrane, and in order for them to function, they need to find maybe a partner protein, and that partner is also associated with the plasma membrane. So it takes that 3-dimensional problem of two proteins trying to find each other that need to coordinate with each other to a 2-dimensional problem, because now they only have to search for each other in 2-dimensional space. So the plasma membrane imposes order there. So imposes order and allows for biochemical activity to occur because you're bringing these biochemically active components into close proximity to each other. Okay, the sixth boundary property that I want to talk about is that the surface of the surface of the cell is where you respond to external stimuli. So the cell is always in contact with the outside world. It's always communicating with the outside world. So cells are going to gather information, and then they're going to communicate that to the inside of the cell. And the way they gather information is through the plasma membrane. There might be receptors on the surface that when something binds, it relays a message to the inside of the cell. You have signaling molecules that do that. When we talk about photosynthesis, photons, of course, are outside of the cell, but it causes a change inside of the cell, so that boundary, that plasma membrane, is responding to those external stimuli. Okay, and then the seventh boundary property that I want to talk about briefly is that this is a place where cells mediate cell-to-cell interactions. And another abbreviation that you're going to see me use throughout the course of the semester for interaction is a lowercase I over a lowercase a. And so this is interaction. And it prevents me from having to spell that out every time. Okay, so of course, epithelial cells form sheets. They mediate cell-to-cell interaction. It's critical that that sheet is maintained because, as you all likely know, if you have a mutation in that causes a disruption of that sheet, that predisposes someone to cancer. And so that sheet being maintained is due to one of these properties of the boundary. Okay, so yes, you're all familiar with the 7 deadly sins, the 7 wonders of the ancient world. Well, herein lies the 7 properties of the boundary. Okay, and these are all outlined for you in your lecture notes as well. All right, so I want to shift gears a little bit and spend some time here talking about the shape, sizes, and surface to volume ratio, and you'll see some homework problems that are related to these issues of shape, cell size, and surface to volume ratio. All right. So this is really a fundamental issue for cells as well as organelles, right? Things that support the inside life of a cell occur at the boundary, right? So if your cell is too large, then you're not going to have enough surface area to support the volume inside. So the smaller the cell, the more surface area per unit volume. Right? And why is this important? The boundary is where everything happens. You have nutrients that need to enter into the cell. You have waste that needs to exit the cell. Heat exchange needs to happen. And so if the boundary is not large enough to support all that metabolic needs of the cell, whatever that is, nutrients in, waste out, release of heat, then that's a cell without a future. Right? So I'm going to start off with one of the most simplistic geometric shapes that we can think about when we start to think about how this size of a cell affects the surface to volume ratio. So I'm drawing a sphere here. We'll give it a radius from here to here. That's R. And the sphere has the lowest surface to volume ratio. And there are cells in the body that have this sort of shape. One in about 50% of the people in this class are oocytes. Also fat cells or adipocytes. And so these are usually just round cells, low surface to volume ratio, very low metabolic needs for these cells, right? Oocytes are just kind of bags of nutrients waiting for fertilization to occur, and adipocytes, bags of fat that store energy. So they, on their own, have very low metabolic needs. But let's go back to this idea of the surface to volume ratio. So let's think firstly about what the relationship is between surface area of this sphere and the radius. And we know that the relationship here is radius squared. And if we look, and many of you probably know the formula for the surface area of a sphere, it's 4 pi r squared, right? So we have that relationship of r squared. And if we look at the relationship between volume and the radius, it's going to be r cubed. And the formula for the volume of a sphere is 4/3 pi r cubed. So if you're tackling some of these homework problems and it's asking you to determine the surface to volume ratio, in no way do you need to calculate each of these equations. You don't need to solve for surface area and you don't need to solve for volume, because what you should do is actually simplify these formulas prior to plugging in. So let me show you what I mean here. So if we're talking about a surface to volume ratio, and we know that the surface area formula is 4 pi r squared, and the formula for volume is 4/3 pi r cubed, right? So that's essentially surface to volume. There's a couple things that cancel out here. The first one is pi's. So we get rid of that messy 3.14 ... All but one of our radiuses will cancel out, right? So we'll get rid of these two and we'll be left with one on the bottom. The 4's will cancel out, and the 3, of course, just because of the nature of where it is going to shift up to the top. So we end up with 3/r. So surface to volume ratio, if you're asked to calculate that and you're given the radius, it's quite simply 3/r. Now, I don't want you to memorize things like this. I want you to actually think about it from a cell biologist perspective. And what does that mean for a cell as far as the radius is concerned, right? There's a super important relationship here. If the radius goes up, so if the radius increases, meaning the cell gets larger, right? So you have a larger number down here. What happens to the surface to volume ratio? The surface to volume ratio actually goes down. So the larger the cell, the lower the surface to volume ratio. The larger the cell, the less surface area per unit volume. So in this case, what we're dealing with is a large cell that cannot support all the metabolic needs because it doesn't have enough surface area. But if we have a cell that has a small radius, so as we lower R, the surface to volume ratio is going to go up. Right? So the smaller the cell, the more metabolic the cell can be. And we can graph this as surface to volume ratio over the radius, and you'll end up with something like this, where the green dot here represents a cell that has a really large radius, low surface area per unit volume, metabolically very inactive. The red dot here, a cell with a very small radius, right? We follow this down here, very small radius, gives it high surface to volume, right? So a lot more surface area per unit volume. Okay, so in super important relationship to remember, and it'll take you through some of those homework problems. What we typically always say is that large cells with a high radius and low surface to volume are geometrically challenged. So they have a geometric challenge. Okay, and of course, again, the size is going to be limited, so you don't just have these enormous cells, right? We're talking about a millimeter being one of the biggest cells. The size of the cell is going to be limited because of the nutrients that need to be taken into the cell, gas exchange, and waste disposal for that cell. But beyond size, I want to think for a minute about shape as well. So that has to do with size, but what about shape? So we talked here about shape of a cell that has the lowest surface to volume ratio, and we see this again for oocytes, adipocytes, just kind of bags of stuff that don't have a lot high metabolic activity. But not all the cells have that simplistic sort of shape. We can go to a few extremes here and think about a neuron, right? Very elongated cell, or a muscle cell from a muscle fiber, also elongated, right? Multinucleated here. So we have a muscle cell or a neuron. These cells are much more metabolically active, so they need a higher surface area. And I'm going to go back to the PowerPoint in a minute to try to convince you that the surface area of this cell and this cell, or the surface to volume ratio, is much higher. So much more surface area per unit volume. So let me take you back to that PowerPoint for a second, and I'm going to skim through a couple of these. We did the same in the in-person lecture. And so the best way that I can think about this is with a balloon. So you blow up a balloon with oxygen from your lungs, and you get this nice, not quite spherical, but assume that it's a spherical shape that I'm showing you on the left-hand side here. The latex is somewhat flexible, just like the plasma membrane could be, right? So what happens if you step on that balloon or you squish it between your hands? Are you changing the amount of air inside the balloon? And you should be saying, no, you're not changing the volume. The volume of that balloon does not change at all. But what you are changing is the surface area, right? The latex is being stretched, right? So you squish that balloon under your foot or between your hands, and you can stretch the latex. Think of the latex as the plasma membrane. And that's the surface area, right? That's the surface area. So the surface area, as the cell goes from this nice cylindrical shape to something longer, like a neuron or a muscle cell, the surface area actually can increase in this situation. No change to the volume, but because the surface area increases, that means your surface to volume ratio goes up. The amount of surface area supporting that volume is greater. You have more surface area per unit volume. Okay. All right. So, great. So this, I want to, move forward now from this and, kind of think about this and wrap your brains around it. But I want to think now about, the different compartments in the cell. So whatever their size and shape, a cell has distinct compartments. And I'm going to define 3 regions of space, and then I want to convince you that 2 of these regions are topologically equivalent. And I'll define what I mean by that in just a second here. Okay, so let's look at these 2 experiments. So we'll follow through on this PowerPoint slide here. So we have a cartoon of the cell, and the 3 spaces that I want to define are the cytosol, marked in red. I want to define the lumen of the organelles that you see here marked in blue, and that's true for all of these different organelle lumens, and the outside of the cell. So 3 spaces that we're going to think about here. All right, so what I want to convince you of in the next few minutes is not intuitively obvious, unless you understand the way that organelle membranes in the plasma membrane behave. And that is that the organelle lumen, so this part here in blue, and the outside of the cell are what we refer to as topologically equivalent. Okay, so what does that mean? It means that whatever is in the outside world can get into an organelle without busting through a membrane. And likewise, whatever is in an organelle can get to the outside world without busting through a membrane. But neither the content in the organelle of the lumen or the outside world can get to the cytosol, at least without busting through a membrane. Okay, so I want to define these 2 spaces with an experiment, right? So let's do that. So let me go back to my notes here. And we're going to look at compartments. And we're going to do 2 experiments. So in the first experiment, what I want to do is inject a cell with a very fine microneedle. I want to inject into the cell a small dye. And then I want to wait a few minutes, and then I want to look at what happens to that dye in the cell. And let's see where that is. So if we were to do this, this is where the dye would be. So we inject it into the cytosol inside of the cell. We wait a few minutes, and this is where it ends up. So where does it end up? It ends up inside the cell, but not inside of organelles. All right. So the dye ends up inside the cell, not in organelles. And this really defines the cytosol. So the region inside the cell that does not contain the organelles. Now you might have noticed, some of you that are observers here, that the nucleus has dye inside of it. And why is that? The nucleus is a very unique organelle. It's not fully encapsulated by a membrane. In fact, it's bags of membranes that are contiguous with the endoplasmic reticulum. So you can see kind of these regions here. And there's pores actually in these regions here. And I'm mucking up this diagram so you can't see it very well anymore, but there's pores there. And by the way, these diagrams are available to you in your lecture notes. But there's pores there where this hydrophobic dye can actually freely pass. So it would absolutely be found in the nucleus. So it is in the nucleus. Okay, so that's the first experiment that defines the cytosol. The second experiment that I want to do doesn't inject the small dye into the cell. Instead, you're going to bathe the cell in media containing the dye. And then you're going to wait a few minutes. You're going to take the cell out of that dye-containing media, and you're going to ask, where did the dye go? And what you're going to find out is it's quite different. So we go back to that cell, we bathe it in dye-containing media. So we'll put this cell in media where the dye is outside the cell, wait a few minutes, wash the dye away, and what we find out is that the dye actually is inside the lumen of the organelles. So the dye is now inside organelle lumens. And we refer to all the organelles that have dye in them as being part of the endomembrane system. And this is almost all the organelles, not all of them. You'll notice here that the mitochondria doesn't have dye in it. So mitochondria is a plastin, as are chloroplasts, and those are not part of the endomembrane system. So we're not going to see the dye inside of these organelles, but all the other organelles. And so this experiment defines the organelle lumens. So these organelle lumens are a place where you can get from the outside without punching a hole in the plasma membrane. And this simple experiment shows us that all these organelles are in communication with each other, and they're in communication with the outside world, right? So how does that work, right? How does dye spread between organelles without breaking through the plasma membrane? So let's look at some ideas here, and in particular, I want to look at budding and fusion of membranes. And we're going to look at 2 examples here that I'll draw out. Similar drawings, again, will be in your lecture notes. So budding and fusion of membranes. And we'll look at this in 2 ways. So the first one is organelle plus organelle fusion. So what I'm going to do is I'm going to draw 2 organelles, and I'm going to use 2 different colors to indicate the 2 leaflets of the plasma membrane. So on the outside here, I'm going to have the external leaflet of these 2 organelles in blue, and the luminal side of the leaflet in red. So out here we have cytosol. Both of these organelles are floating around in the cell, and then we have the lumen inside. And I'm going to draw some contents in here just as these little dots here, because these organelles obviously have stuff inside of them. And then I'm going to draw on one of them a protein that has different ends, so that we can see what happens to the contents. We can follow the lipid composition, and we can follow the protein when we have fusion occurring. So this is fusion. So when these 2 organelles come together, and I'm going to draw it like they kind of just did, and their fusion is not complete, what we maintain here, and this one might be somewhat intuitive, is that the membranes facing the cytosol after fusion are still facing the cytosol, and the membrane facing the lumen is still facing the lumen, the leaflets. So that orientation is maintained. So orientation maintained. And this is for the lipids, right, in the 2 leaflets. The protein is also going to be maintained in its orientation, where the region out here that was associated with the cytosol prior to fusion is still associated with the cytosol after. So we maintain the orientation of the protein. And again, that's going to be a P, capital T, or sorry, capital P with that little hash that looks like a T there. So we maintain orientation of that. And then what happens with the contents is the contents of the 2 would get mixed. Okay, so we have mixing of contents. Okay, and I'll just take you up briefly to the diagram that's included in your lecture notes, kind of here, very similar. We have fusion, and the part of the protein associated with the lumen is still associated with the lumen. The part associated with the cytosol is still associated with the cytosol, and you maintain orientation of those 2 leaflets of the lipid membrane. Okay, so that's the case for organelle fusion. What about an organelle to the plasma membrane fusion? Let's look at that. So organelle plus plasma membrane. I'm going to draw a cell, and I'm going to be consistent to my upper diagram where I have the blue leaflet facing the cytosol. I'm again going to have a blue leaflet facing the cytosol, and my red leaflet for the whole cell will be facing the outside world. So here we have a cell, and inside of that cell, we have an organelle. Blue leaflets both facing the cytosol for the organelle as well as for the plasma membrane. We have some cargo inside of here that we want to follow where it's going. And again, we want to look at the orientation of a protein. So let's go ahead and put a protein in here. Okay, so what happens here is that we have fusion, of course, right? But when we have an organelle fuse with a cell, we also can refer to this process as exocytosis. But for today, our purpose is really to think mostly about what's going on here. And so I'll draw this out in a second in a new diagram, but what's ultimately going to happen is when this organelle fuses with the plasma membrane, the blue membrane is going to fuse with the blue membrane. So the two cytosolic faces will fuse, and the luminal leaflet will fuse with the extracellular leaflet of the plasma membrane, such that if I were to redraw this out, it would look like this. So we have in red, again, the leaflet facing the extracellular space, and in blue, the leaflet facing the cytosol. And the contents, of course, are going to be released from the cell. And the protein also doesn't change orientation again. So what we have here is a portion of the protein that was associated with the cytosol stays associated with the cytosol. The membrane that was associated, we'll just go with blue because that's what we have, the membrane associated with the cytosol, whether it was in the organelle or, of course, the cell, when the organelle fuses to the cell itself, to the plasma membrane, that leaflet is still associated with the cytosol. Okay. All right, let me, and then importantly, of course, the contents are secreted. You can run each of these processes in reverse. So this is fusion going down here. If we run it upwards, this would be budding, right? So you can start from the larger organelle shown on the bottom here, and it can bud off into two smaller organelles. Similarly, in the second diagram, instead of looking at exocytosis, if we went the other way, it'd be endocytosis. Right? So you can have a organelle created off of the plasma membrane. So let me just finalize this by showing you this on the lecture or on the PowerPoint slides. These are the diagrams in your lecture notes. So this is organelle fusing with the plasma membrane, and you'll see same thing here. So we've maintained orientation of the protein. The side of the protein that was in the lumen of the organelle is now outside of the cell. It'll be really important as we progress through this course to understand this concept. Side of the protein in the lumen is now on the outside of the cell. The side that's facing the cytosol is still facing the cytosol. Okay. All right, so a couple comments here. The contents are secreted. They become extracellular, so they're no longer inside the cell. And the cytoplasmic leaflet and cytoplasmic portion of the membrane protein remains cytoplasmic. I already mentioned that. That's highlighted in red here. I just circled that. And the luminal leaflet and luminal portion of the membrane protein becomes extracellular, and that I will now circle here, right? All right, so we're going to end for today. Again, my apologies to all the IT issues. I'm doing my best to try and keep us up to speed here, and my hope is that we are back on track for Friday. Enjoy your evening. We'll see you soon.